Okay, so now we will be talking about cell division, mainly cell cycle. I'm going to set up the screen so I can draw, and then we can follow along with the PowerPoint down below. Now, what is cell cycle? We're mainly talking about how cell is always in its life at certain stages, and that's called a cell cycle. Like we're either in the spring, in the fall, in the summer, or in the winter. Same thing for cell. The cell is always in a certain stage. But why do they go through these cycles? Because they can then grow one into two. Okay, so cell cycle is different stages for cell to ultimately one become two. Now there are these 5 phases in cell cycle. Okay, so the first phase is called the gap 1 phase, G1. This is usually for the cells the longest phase. Okay, this is where the cell is growing. Okay. And then once the cell have obtained a lot of nutrient, enough energy, enough organelles, and various different cellular components, it can now say, I want to one become two. I am ready to go on to the next stage. When a cell wants to go from 1 into 2, it then needs to synthesize its DNA. So it has 2 copies of its most important genetic content. This way, when it splits, each cell can have 1. That phase is called S phase, which is also called synthesis phase. So what happens in the synthesis phase? That's when DNA replication happens. We talked about this way early on. We talked about how DNA synthesizes itself. Remember the replication fork we drew out? That's DNA replication. So in synthesis phase, one strand of DNA is going to have an identical twin mate, identical. If you say A, I also have a strand that says A, and then T. G, and then C is identical. Okay, so G1 longest, then S to replicate DNA. And then before the cell goes into active splitting and everything, goes into another gap phase called G2. G2 is when organelle replicates, microtubule organizes, and we're going to talk about microtubules. They are the main movers once you go into the final thing, which is called mitosis. So I'm going to extend my G1 because G1 should be way longer. And then now we are in M phase. M phase is probably what you all have learned back in high school. Okay, you got the 5 phases: prophase, prometaphase, metaphase, telophase, and then, anaphase, and then telophase. Okay, so these are the main 5 phases that will go into painstakingly very detailed. But zoom out. All those 5 phases is part of the mitosis. Okay, and then all these 3 phases together, G1, S, G2, are then called interphase. Okay, there you go, interphase. Finally, when the cell finally reaches the end of mitosis, it then splits into 2 cells, which both cells can then circle back to G1. Each of them can then go G1, G2, and then split again. And this process of one splitting into 2 is then called cytokinesis. So it's the active breaking of the two, okay? Okay, so we'll move on from cell cycle. Just know that with different cells, it takes different time, okay? Bacteria doubles way faster than we do. They can do this every 20 minutes. Mammalian cells, usually 24, 48. I played around with the mammalian cells that actually takes 72. So it really differs. Now, that's the picture from your textbook, and it clearly shows regarding interphase, M phase, which have mitosis and kinesis. Okay, so interphase contains G1, S, G2, and then we'll later touch upon how there's a structure on your chromosome called the centromere. Okay, so this is your chromosome. Like I said, chromosome is highly condensed, right? Because you're ready to move. That's what happens in mitosis, or actually when you're getting ready for mitosis, you then highly condense. That's going to happen in prophase. In the middle of that is called centromere. And centromere actually is attached by protein in the blue called kinetochores, okay? And then remember how during S phase, you then make sister chromatids. So they're identical. That's why there's two. And then ultimately, there's going to be strands of microtubules that comes in and binds to the blue part, which is the kinetochore. Why? Because then we can move the structure around, okay? Now, let's move to the next slide. Okay, now let's talk about the interphase. So we're not at mitosis yet. We're just talking about during G1, during G2, okay? What happened is you're trying to replicate DNA or you're trying to grow. Both things, you need your big book open. Your DNA needs to be open so I can replicate, I can read. Your big book needs to be open. Your DNA needs to be in a decondensed. When I say open, I mean a decondensed structure. If you're tightly bound, that's a closed situation. Open, so then if you're growing, I need recipe. I need to make protein for you to grow. If you're replicating, I need access so that I can copy. So what is it going to look like in the cell during interphase? This is the cell membrane. That's your nucleus. Your DNA is not going to be highly compacted. They're all very much dispersed. So you can't really tell, it's not gonna have a really strong structure, okay? But right before you go into M, okay, the cell is going to replicate something called the centrioles, as you can see right here, centrioles. Usually the cell should only have, okay, the centrioles usually look like this. They're like a T structure. And then they can send out microtubules. Okay, that's what I was drawing. But then they will replicate. Because why? Now they need to move along, okay? So that is your interphase. Now we're going to move into the actual mitosis. And yes, please draw with me. Have a piece of paper, have beautiful colors of various different pens or pencils, and we're gonna draw the 5 phases. First phase. So you gotta know how to, the name of the 5 phases, and you have to know what's going on with the chromosome during each phase, what's going on with the nuclear membrane, what's going on with the centrioles and microtubules. Okay, these are the main players. Now let's start with prophase. So let's write prophase. Please know how to spell, okay? And then you have to draw out the cell membrane. So know that's the cell membrane. I'm gonna keep that all in black, okay? So what happened in prophase is because you just copied your DNA and they're still loose, but I want to move them in mitosis. I want to move them into 2 separate cells. So I have to first condense my chromosome right here, condense chromosome, okay? So that's the first thing with prophase. Condense chromosome. Nope. Chromosome. Okay, so you want to condense your chromosome. Okay, and another thing is the chromosome is actually inside the nucleus. Okay, but remember I said, look at this, your chromosome is inside, like in red, right? So your chromosome is inside of what? This green thing. What is this green thing? Your nuclear envelope. Now, who's the mover? Who did I say was the mover? Okay, I really should have drawn that in yellow because guess what? That's this. Okay, let me just redraw that. I feel like that might help color code everything. Okay, so the yellow is actually just these, okay? So these are gonna reach inside and actually move these structure. But like I said, in interphase, it's dispersed. But during prophase, it's going to condense. Okay, so let's draw this out. I'm going to keep the color. So I'm going to keep the chromosomes red. I'm going to keep the nuclear envelope green, and then my microtubules yellow. Okay, so let's try this. First of all, my chromosome needs to condense. So let me draw 2 pairs of homologous chromosome. Let's think about it. 2 pairs of homologous chromosome going to mitosis. Pairs, okay? So if we started with, oh, I said red, okay. That's 1 chromosome, right? And it has a pair. So they are, let's say they are a pair. But during S phase, what happened is they're going to synthesize, agree? Does that make sense? So they're going to look like this when they enter prophase. There's going to be 2 of every chromosome, and then that's 1 pair, okay? You know what? I'm just going to do 1 pair. I think that will make things easier so you guys aren't waiting for me to draw. So this is 1 pair of chromosome going through mitosis. Now they're going to condense. So the first thing, chromosome is condensed. That's the first thing right here. Condensed chromosome, check. We already did that, okay? And then spindle apparatus, what is that? Okay, that is the very microtubule we talked about. That was the yellow drawing that I just did up here, okay? The 2 centrioles is going to move to the opposite pole and form spindle apparatus. That means you're going to have, I'm not going to draw it all the way because it's in the process thing, right? It's going to move from right next to each other to start moving all the way around. So it is an active thing. It is moving, but it's going to send out its what we call spindle fibers, okay? What's trying to do is this thing. It's trying to gonna try and attach to your chromosome. Yet, guess what? Your chromosome lives in your nucleus. So, third thing, nuclear envelope needs to break down because then my cables can reach in and actually grab on. So, this is prophase. First, your chromosome needs to condense. Second, your spindle apparatus assembles and move towards opposite poles because it's kind of hard for me to write all those points on the iPad I'm using. This pen really is a bit too thick for like fine writing. So please use my guide down here, but you do need to know how to spell out the name and 3 main points that happens. Okay, that is something that you want to list in your drawings, and then you have to draw it out. So this is what's happening in prophase, and then we're going to move and you can see. So here we go. What did they draw out? They actually drew out 2 pairs of homologous chromosome. I only drew out 1 pair. Okay, they have 2 pairs. I would say this long blue and this long red is 1 pair. This shorter blue and this shorter red is another pair. So they have 2 pairs of homologous chromosome. And each chromosome currently have a sister chromatids. Okay, that's what you're looking at. And this green thing they're drawing out is what I was drawing out here in yellow. Okay, okay. And then obviously they listed everything. So I'm just really breaking down everything you can find on my PowerPoint and talking them through one by one. That's prophase. Let's move on to prometaphase. Okay, so again, let's spell out together on your paper, prometaphase. Okay, and then prometaphase also still have the cell membrane. Okay, but now the cell is ready to transition. Okay, transition happens after disassembly of nuclear envelope. Now what's going to happen is the nuclear envelope is gone. So you're not going to see me draw out anything green at this point forward for a long time until we're in telophase. Okay, and then the main thing is that microtubule is now going to attach to the chromosome. It's ready to latch on to the kinetochore and move it around. So it will now be an opposite pole because they accomplished that. And then you're going to have microtubules reaching across, okay, to kind of set up the spindle. And then it's going to try and reach out to do what? It's going to try and catch the chromosome. Okay, so what I just drew out in yellow is the microtubule. And you have to recall back, we talked about this in chapter 4. It is the cytoskeleton. And then we're going to draw out our chromosomes. Okay, and so this cable latched out and then actually got the chromosome. Okay, in the middle centromere, and I'm going to just dot the kinetochore right there. Okay, remember the blue is like made of protein kinetochore, where the yellow will then latch onto. What about the other one? The other one is also, okay, is also now attached. So what did I just draw? Microtubule attachment. Okay, you got groups growing from the poles. So you got these coming out from the poles. These are the poles. That's what they're talking about. And each chromatid, okay, needs to now be connected. So this one needs to connect, okay, and this one needs to connect. So now they are connected. Why? Because ultimately, once, for example, I am the chromosome, I got this be the chromosome. I got spindle fiber coming out, attach. Spindle fiber on the other side, attach. Okay? And then now I can push using my spindle, push over, and I can pull and push this thing to do what? To eventually align all the chromosome in the middle, and we'll talk about why. Okay, so prometaphase is when you got the envelope actually gone and the spindle fiber began to reach out and latch onto the chromosome, and the process of chromosome actually beginning to move towards the middle. That's prometaphase. Okay, now as you see here, this is a better drawing, but I'm just kind of breaking it down step by step. Then we move on to metaphase. Let's spell it together, metaphase. Metaphase again has you're still inside a cell membrane, okay? And what happened is you are seeing actual chromosome right here, pretty cool. That blue part is all the 46, if this is human, chromosomes lining up in the middle. And then this red thing is the microtubules that's reaching out. So what happened in metaphase is you got the centrioles coming out. Okay, and then they're going to do this. And some of the microtubule just overlaps. Some are gonna then attach to your chromosomes and make sure the chromosomes aligns in the middle. Okay, so let me just draw that really precise. This is supposed to hit the middle, that's supposed to hit the middle, this is supposed to hit the middle, this line should hit the middle because that's where the centromere is. And both chromosome needs to line up at the middle plate. Alignment of chromosome along the metaphase plate is what happened at metaphase. That's the third phase. There we go. Down below, there are 2 pairs of homologous chromosome. I am drawing out 1 pair of homologous chromosome. The main thing that happened is chromosome all line up in the middle. Now let's talk a little bit about why. Okay. Eventually, the cell wants one of everything inside a cell. Okay. 1 of this chromosome, 1 of this chromosome. Okay. So that means, how does the cell know that it didn't just take randomly 2 chromosomes in the end? What if I took 2 of these instead of 1 of these and 1 of these? That would be terrible because I have 2 sets of everything, but I don't need that. I only need that 1. What am I saying? I am saying, okay, human have 46 chromosomes total, right? But I want 1 of each of that 46. Imagine our classroom. Let's say we have 46 students in there and Dr. Yang. We want to copy our classroom. So imagine the class as a cell and students and me as the chromosomes. If we want another class with one another cell exactly the same, we'll replicate. I will get another Dr. Yang. Okay, student A will have another student A. Everybody will have a clone. And then now we're going to move half of the class over to the other class. But what if we just randomly sent 46 people over? And that class end up having 2 Dr. Yang. And there's no Dr. Yang in this class, but there's 2 student A. That's not copy. But how does the cell know how to retain one of each? So the cell's very smart. The cell says, when you copy, right? So like this particular chromosome, when it copies, it's going to hold on to its twin. Like Dr. Yang is going to hold on to her clone, okay? I'm not going to let go. That's why you see them together the whole time until we all line up in the middle of the classroom and we're holding hands with our clone. Then it's clear. There's one Dr. Yang on this side, on the left. There's one student A on the left, one student B on the left, one student C on the left. Same thing. One Dr. Yang, one student A, one student B, one student C. That will then be clear right in the middle. What do we need to do? Then we just split it in half in the middle, and then we'll get one of everything without problem. That's usually when it goes right, okay? So we'll talk about if it goes wrong, then we'll have a genetic problem, right? But most of the time, the cells can do this faithfully. So this meta plate alignment is super crucial. Cells usually check and check and check and make sure that they've got all the twins line up faithfully, or else they're not going to break them apart, because then it's not going to be genetic even. We'll talk more about this, and this could potentially become a problem if cells aren't following what they're supposed to do. Okay, so super important metaphase. We're on our 3rd phase. We're moving on. This structure is exactly what happens. You got microtubules, which is this one, coming in and grabbing on. You got the other side coming in and grabbing on. And then you actually have sister chromatids, right? So they're grabbing on and they're all aligned. And eventually next, Anaphase, the 2 sisters are going to then split, because one Dr. Yang and clone Dr. Yang split. Then each classroom gets one Dr. Yang, or each cell gets one chromosome 1. Okay, so that's coming up. Anaphase. Let's draw Anaphase. Spell Anaphase. Okay, what's happening in Anaphase? The centromere splits, the twin splits, and move to the other side. So sister chromatids, remember they're identical, they're called sister chromatids. They're pulled what? Towards opposite pole. Okay, so let's look at that in a picture. Oh man. Oh, come on. Okay, so like so. So let's draw this out. What do you mean? Okay, so I got my spindle fibers, and again, they're sending out, but they're trying to separate now. Okay, oops. How did that become? Okay, and so what happened over here? Do you see that red? They're now going to go opposite. One's going to go this way, the other one's going to go that way, the one's going to go this way, one's going to, okay, so let's draw this out. Now I have one going this way, the other one going this way, and one goes this way, and the other one goes this way, and this one is actually, okay, so it's getting pulled in the middle. That's why it has this structure, right? So it is kind of like think of it kind of floppy, and as you pull it in the middle, the 2 ends are going to drag. That's why we draw it out that way. That's why these are drawn out that way, because originally they're stuck together in the middle, and when we break them and start to drag them, then it starts to look like this. Okay, so what's the main thing that happens in anaphase? Protein holding centromeres are degraded. Originally they're held together, but like I said, once you have them line up nicely and we double check, we have one over here, one over here, everything is one on each side, then we can pull. Then we ensure we're going to replicate two cells that's identical. Finally, we're going to move to telophase. Last phase of mitosis is when spindle fiber disassembles and nuclear envelope forms and chromosome begin to uncoil. Okay, so it's everything opposite of prophase. Prophase, three things happen. Telophase, three opposite things happen. Prophase, chromosome condense. Okay, chromosome condense, that means telophase, chromosome begin to uncoil, decondense. Nuclear envelope dissolves, that's the second thing. Nuclear envelope begin to appear. So everything opposite, because now I'm ready. Originally, spindle apparatus assemble, now it's going to disassemble. Okay, so everything just the opposite. That means what? My spindle fiber is going to become shorter. Okay, I really shouldn't be drawing out telophase like this. Let me draw again, because really they're becoming two cells. So that wasn't a good portrait. I'm going to kind of make them kind of two cells. And they're now almost on the opposite side, okay? And they are beginning. Okay, so this is one, this is one, this is one, this is one, oops. And then what's going on? The nuclear envelope is coming back. Okay, so we can see that here. Let me see. Look at that. What is this blue haze stuff? That's your nuclear envelope coming back. Your microtubules are still in the middle trying to push each other towards the opposite pole. Your chromosome, again, this picture, there are two pairs of homologous chromosome. I am only drawing one pair. You have a total of 23 pairs. So on the exam or something, I can be talking about different pairs, and you need to be able to kind of follow and understand what I'm asking. Okay. So what happens in telophase is chromosome began to decondense. I didn't draw that out, but know that's everything opposite of the prophase. It will start to decondense, nuclear envelope reforms, okay? And then spindle fiber disassembles. Okay. So those are the 5 phases of mitosis. And then last, they're not fully separated, right? Last is called cytokinesis. Let's all cytokinesis. Let's all write that out on your notes. Cytokinesis is the very end. The two cells then separate. Look at this, pretty cool, right? This is cytokinesis. You have actual split of the cell membrane. Okay, so that's mainly it. Okay, I'm gonna make another video about cell control, which is really cool. This will be talking about cancer. Finally, we get to talk about something really cool, something we really care about, and you can apply most of the stuff we talked about. We will be talking about cell cycle and why that's important. There's checkpoints, okay? So kind of review, definitely hope you're able to draw out what is a cell cycle and be able to name the phases. Know what is interphase. It contains G1S, G2. Know during interphase, your chromosome is not condensed, so they kind of look like spaghetti, they're opened. Then at prophase, it condenses, and that's when you see the structure, and you see 2 of them because the cell has gone through S phase, which means it has synthesized an identical clone of itself, and I am drawing out 1 pair of homologous chromosome that each have its twins, and they're holding hands. So there's now 4 chromosomes on here, mainly because there's sister chromatids, and then they're a pair. Okay, and then Prometaphase, Metaphase line up in the middle. Anaphase, they split. Telophase, not that one. They finally are going to each end, and then cytokinesis. Okay, make sure you draw this out. And if you like, you can show it to me through Slack or email it to me.